So why don't we move on to our next speaker, who is the recipient of the Rising Star Award. So the Rising Star Award is now in its third year, and it's what various reviewers considered to be the best abstract submitted by an emerging San Diego CFAR researcher. It was first introduced in 2018, and the recipient receives a commemorative plaque, a $200 allowance for research-related purchases at the UC San Diego bookstore, and much admiration. The first year's honor went to Sarah Gianella Weiwel for her abstract on compartmentalized HIV rebound in the male genital tract after ART interruption. And she's gone on to do great things and is now the director of our translational virology core, and also the recipient of a prestigious NIDA Avenir Award, an R01, and most recently is a co-PI on the T32 HOPE training grant. The following year, the award was presented to Sunny Yo at the Sanford Burnham Prebys Medical Discovery Institute for her abstract on CGAS sensing of HIV infection initiated by the QBP1 recognition of the intact capsid of incoming virus. And we're pleased to note that her abstract was based on her 2017 CFAR Developmental Award. And she's still publishing on this topic. As you can see, the Rising Star Award has gone to talented emerging investigators who've continued excellent work. And we know that the recipient of this year's honor has a similarly bright future. So we're delighted to announce that Dr. Marva Seifert is the 2021 Rising Star for her abstract, entitled Novel Bioelectronic Assay for the Detection and Quantification of Mycobacterium tuberculosis antigen, CFP10, in serum and urine for rapid diagnosis of active TB disease. We're even more pleased to note that this abstract and the presentation you're about to hear resulted from yet another CFAR Administrative Award. This is the CFAR Administrative Supplement, which Dr. Seifert received on point-of-care electrochemical immunoassay for TB diagnosis. She is assistant professor in the Department of Medicine at UC San Diego, focusing on tuberculosis research, and which we all know is a serious co-infection for people living with HIV. She's primarily focused on the development of and an evaluation of rapid diagnostics for pulmonary infectious diseases, specifically TB and associated drug-resistant variants. She's particularly interested in leveraging novel technologies to develop innovative point-of-care solutions for traditionally difficult to detect pathogens. And most recently, her work has focused on the uptake of novel SARS-CoV-2 diagnostics in underserved communities. So congratulations from all of us at CFAR to Dr. Seifert. And we're looking forward to hearing your presentation and following for your future success. Dr. Seifert.
Thank you so much. I'm very grateful for the award and excited to be part of such an illustrious group. Thank you for the opportunity to present results from the evaluation of our novel bioelectronic assay for diagnosis of active TB. First, I'm going to very briefly touch on global TB and the tools that are currently used to diagnose active disease, just to provide some context for our assay development. Then I will introduce you to the technology that we used for the assay. And then finally, I'll present the results that we obtained using clinical samples, which of course is the most exciting part of this presentation. As you know, TB's been around for millennia, and the untreated case fatality rate before the advent of current treatment regimens was estimated to be somewhere between 50 and 70%. TB is primarily a pulmonary disease. However, it can infect other tissues and organs, and about 15 to 20% of all cases globally are actually extrapulmonary. And in the context of diagnosis, assays optimized to detect Mycobacterium in sputum may be less sensitive in other body fluids, increasing the chance of misdiagnosis for individuals with extrapulmonary disease. More adult men than women are infected with TB. However, the male to female ratio varies by prevalence of HIV infection. And in regions where HIV is highly endemic, the male to female ratio for TB disease can equal 1. Currently, the World Health Organization estimates that 10 million individuals develop active disease each year, and 12% of all cases actually occur among children less than 15 years of age. About 1.4 million deaths occur each year due to TB, and 0.2 million of those occur among individuals living with HIV. So both children and individuals with HIV actually make up a pretty significant proportion of TB cases. In 2019, only in the last year that we have data, only 57% of all reported cases of disease were actually bacteriologically diagnosed. Diagnosis without bacteriological confirmation can lead to an initial misdiagnosis and unnecessary treatment, or could lead to delay in treatment, which may result in an increased risk of poor treatment outcomes. The World Health Organization estimates that there is a gap of approximately 3 million cases between the number reported and the total number of cases globally. And this is due both to some underreporting and primarily to misdiagnosis. This significant gap in misdiagnosis is due in no small part to the lack of a highly accurate, rapid, and fundamentally inexpensive point-of-care diagnostic tool for TB diagnosis. So what are the tools that are currently available for diagnosing TB globally? One of the original diagnostic methods, imaging or X-ray, was based on detection of abnormalities of the lung and was relatively sensitive. However, it was not particularly specific. Sputum smear microscopy, again, a method that was introduced over a century ago, is actually the most frequently used diagnostic tool for active TB detection in many endemic regions. And this is in no small part because it is inexpensive and rapid. However, it has very limited sensitivity, even among immunocompetent individuals, and poor sensitivity among people living with HIV, less than 50% sensitivity, and even lower sensitivity among children. Other diagnostic solutions include culture-based methods, which are actually highly sensitive and specific, but they can take weeks to produce results, and they require complex laboratory infrastructure and biosafety capacity. So this reduces their utility as a point-of-care diagnostic. The advent of novel molecular-based techniques has changed TB diagnosis in high-income countries. In particular, Seifert Gene Expert has significantly changed kind of the diagnosis landscape globally. However, even with its price adjustment for low and middle income countries at 10 to 30 US dollars per test in the public sector, is actually still cost prohibitive as a screening tool for some national TB programs. And so it tends to be reserved for detection of resistance or use among high-risk individuals. Its cost in the private sector, even in low and middle income countries, actually easily exceeds 100 US dollars. So these issues are actually similar across other nucleic acid tests, particularly the higher complexity assays. There are some antibody detection techniques such as quantification, which use a slightly different method to detect TB infection. They stimulate an antibody response ex vivo to TB antigens. However, these tests actually can't differentiate between latent and active disease, so again, are not useful as point-of-care diagnostics. And just to note, most of these commercially available molecular-based diagnostic tests are optimized for sputum as a clinical matrix. And so this, of course, reduces their utility among patients that have trouble producing sputum, and that includes children and individuals living with HIV. It's also, of course, the detection of extrapulmonary disease, as we mentioned earlier, is also severely limited by sputum optimized diagnostics, as the sensitivity of these diagnostics solutions is highly variable in non-sputum sample types, even, for example, the Cepheid gene expert test. There is a promising alternative, however, with antigen-based assays, which detect circulating Mycobacterium tuberculosis specific antigens in easily sampled body fluids, such as blood and urine. Commercially available urine-based antigen diagnostic assays have demonstrated relatively moderate sensitivity among immunocompromised individuals, which is fantastic. However, these assays really have continued to demonstrate poor sensitivity in the general TB population. There are, however, some research-based assays for detecting Mycobacterium tuberculosis antigens in both urine and serum that have been very successful at identifying patients with active TB. But due to their technological complexity or costly instrumentation, these research-based tests have not been translated into point-of-care diagnostic solutions. So our goal was to build on the success of these research-based antigen detection assays and develop an assay that could detect CFP10, MTB-specific antigen, in circulating body fluids, both to reduce the dependency of TB diagnostics on sputum production, and also to make an assay that would be hopefully more sensitive in detecting extrapulmonary disease infection in children and in people with and among people living with HIV. We selected CFP10 as our target antigen due to its relative specificity, as it's only present in one non-tuberculosis mycobacterium that causes human disease, M. kansasi. CFP10 is also excreted by the bacteria, and so we anticipate that it would be more likely to be detected during active disease, during bacterial replication of active disease. We partnered with Joe Wang in his laboratory that had already successfully optimized a technology that could detect ultra-low concentrations of enzymes and antigens in small volume, unprocessed clinical samples. This technology was an inexpensive bioelectronic sandwich type assay that used a sensor that was fabricated from inexpensive plastic substrate, which had 2 electrodes on the sensor, a reference counter electrode and a working electrode. Additionally, a commercially available assay reader could be used as it could be used to read these sensors, and so that really reduced kind of the need for a specialized equipment at point of care, or to make the test itself much more inexpensive. So the results I'm going to talk about from here on out were just published last week in Scientific Reports, and the figures in this presentation are mostly adaptations of the figures presented in the publication. So here's a schematic of the final prototype assay. After fabrication and preparation of the sensor, the capture antigen CFP10 specific antibodies are immobilized on the working electrode, and at this point, the sensors can be stored for future use. 10 microliters of serum or whatever sample matrix you plan to use is then deposited on the modified working electrode and incubated for 45 minutes. This binds the CFP10 antigen to the capture antibody. After a washing step to remove any extra sample matrix and to stop the immunoreaction then occurs, and then 10 microliters of horseradish peroxidase, HRP, that's horseradish peroxidase, labeled anti-CFP10 detector antibody solution is then placed on the working electrode and incubated for another 45 minutes. And this essentially sandwiches the CFP10 from the clinical sample between the capture antibody and the detector antibody on the working electrode. The sensor is washed again to remove any extra HRP detector antibody, and then the sensor is inserted in a potentiometer that's attached to a laptop in our case, and a 20 microliter drop of tetramethylbenzidine hydrogen peroxide is placed over both electrodes, and a current of negative 0.1 volts is applied, and the ampermetric signal is recorded for 2.5 minutes. The current measured from the potentiometer corresponds to the amount of detector HRP tag detector antibody that's bound to the sensor. And this, of course, corresponds to the concentration of the CFP10 in the sample. And so this is how we're able to quantify CFP10 in our clinical samples. The figure on the right shows how if there is no HRP tag detector antibody bound to the sensor, there'll be no difference between the working electrode, which is the purple signal, and the reference electrode, which is the black dashed line signal. However, if there's HRP tag detector antibody bound to the sensor, you can see a signal difference between the reference electrode and the working electrode, between that blue TB infected signal line and then the dotted black line. So after multiple optimization steps, of course, which I will not describe here, amphiometric responses to varying concentrations of CFP10 spiked buffer, urine and serum, were actually used to construct calibration curves to establish matrix-specific baseline analytical performance. The figure on the left is the initial calibration curve in buffer using spiked CFP10 in concentrations ranging up to 250 nanomolars. Or nanomolar, excuse me. The figures to the center and the right are the calibration curves for urine and serum respectively. CFP10 standard curves, or standard, was spiked into human serum and urine to achieve concentrations that range from 0 to 25 nanomolar. Based on these calibration curves, you can see analytical characteristics of the optimized assay in the table in the upper right corner for the different matrices. The estimated limit of detection of CFP10 antigen was 0.7 nanomolar in urine and 0.4 nanomolar in serum. So that's essentially 44 picograms in a 10 microliter clinical sample droplet. We evaluated the reproducibility of the assay by running 6 optimized sensors using a positive control of 100 nanomolar concentration of CFP10 antigen spiked in buffer and 6 negative buffer controls. You can see the results in the figure on the left. The relative standard deviations for the positive and negative controls were 5.9% and 5.2% respectively, indicating relatively good reproducibility of the assay. The sensor's stability over time was assessed by evaluating prepared sensors, so sensors that had the capture CFP10 antibody immobilized on the working surface, that had been stored at 4 degrees over the course of 30 days. Amphiometric responses using positive controls ranged from 228 nanoamperes to 247 nanoamperes, and no systematic decrease was observed, as you can see in the figure on the right, indicating relatively acceptable stability over the 30-day observation period. Our next step was to evaluate the performance of the assay using clinical samples. Clinical samples used in this study were obtained from 3 independent cohorts, 2 previously completed studies, and 1 ongoing pulmonary TB study. Samples were collected under each study's respective UCSD IRB approved protocol and had been biobanked for future use. Informed consent was obtained at the time of specimen collection from all study participants. Participants in cohort A were enrolled in an antigen diagnostic study here in San Diego. Both serum and urine specimens were collected from patients presenting for acute care at UCSD hospitals and who were assessed for TB using culture and/or gene expert. Due to some enrollment delays, however, some of the TB positive participants in this cohort had initiated treatment prior to our sample collection. Participants in cohort B were recruited in Moldova as part of a study designed to identify blood-based biomarkers of progression from non-infected to active TB infection. Serum samples were collected from index culture positive TB cases at treatment initiation. And again, some of the samples were actually collected just post treatment initiation. There were also samples that were collected from close contacts of index patients who were determined ultimately to be TB negative. Participants in cohort C were also enrolled in a Moldovan study, which was designed to evaluate a novel molecular diagnostic for drug-resistant tuberculosis. Study participants consisted of individuals who were considered at risk for drug resistance, so they were actually confirmed TB patients at the time of enrollment. And in this cohort, initial serum samples were collected at enrollment, so before treatment for their drug-resistant episode was started, and then again at predetermined set intervals throughout treatment. The biobanked serum samples and urine samples were stored at -20 degrees Celsius for a minimum of a couple of weeks to a maximum of 3 years. prior to being used for our assay evaluation. You can see in the table on the right, the mean age of the participants was 42 years, about 84% of them were males, and 2 individuals were living with HIV. So in total, we evaluated 33 serum and 10 urine samples from patients at risk for active pulmonary TB or drug-resistant disease. All samples were run in triplicate, and results include standard error bars. Here you can see the raw amperometric responses for each sample evaluated. The serum results are in the top figure and the urine results are below. The initial white bars represent serum and urine negative controls that were commercially purchased, and clinically and bacteriologically negative participants are represented by the maroon bars. Participants who were TB positive and untreated are represented by the blue bars, and those who were TB positive and on treatment at the time of sample collection are represented by the purple bars. The number of days a participant was on treatment prior to sample collection is indicated by the underlying number above the bar and ranged from 1 to 17 days. You can see that none of the clinically or bacteriologically TB negative participants had amperometric responses above the negative control line. All untreated TB positive individuals and those who had less than 1 week of treatment produced amperometric results above the negative control line. Participants with bacteriologically confirmed TB, but who had been on treatment for a week or more, produced somewhat variable amperometric responses. So this translates into a sensitivity among participants who were untreated or treated for less than 1 week at the time of collection of 100% with a confidence interval of 81 to 100 and a specificity also of 100% with a confidence interval of 72 to 100. And just a note of caution, of course, that we did, this is only, from a limited number of samples. The aggregated mean and interquartile range of amperometric signals are presented in the box and whisker plots below. And the plot on the left represents the clinical serum samples. And the figure in the middle are urine results. So the results are grouped by TB diagnosis and treatment status. The mean amperometric serum signal for TB negative participants was 41 nanoamperes, and for TB positive untreated participants, it was 77.6 nanoamperes. And for TB positive treated, it was 61 nanoamperes. So you can see that there's a really good separation between TB negative participants and TB positive untreated participants. Using the concentration curves that we described earlier, this translates into CFP10 concentrations of 1.1 nanomolar and 5.9 nanomolar for TB positive patients with less than 7 days of treatment. And for patients with more than 7 days of treatment, CFP10 concentrations were mostly undetectable. The mean amperometric signals produced by urine samples were slightly higher than the serum results. Average amperometric response for TB negative individuals was 53 nanoamperes. For TB positive untreated individuals, it was 156 nanoamperes, and for TB positive treated individuals, it was 82 nanoamperes. The correlation of amperometric responses obtained from paired serum and urine samples from the same participants are presented in the figure on the right. Estimated CFP10 concentrations in urine appeared to be up to 2 times higher than the matched serum samples, indicating that our assay might ultimately have greater sensitivity for identifying TB cases using urine as a clinical matrix as compared to serum. And this actually is pretty consistent with other TB antigen studies that have evaluated concentrations of Mycobacterium tuberculosis antigens in TB patients, which have also reported that the higher antigen concentrations in urine compared to serum. So the variability we observed in CFP10 concentration among treated individuals led us to evaluate some serial samples from 8 drug-resistant TB patients in study cohort C, in order to determine if there was maybe some association between CFP concentration and treatment response. All 8 patients were sputum smear and culture positive prior to treatment initiation. Follow-up samples were collected between 1 and 3 weeks after treatment initiation, and follow-up sample collection time At the time of follow-up sample collection, 5 of the patients were smear negative and culture negative, and 3 were smear negative but were still culture positive. So in the top figure, the amperometric responses from patients on day 0 before treatment initiation are represented by the blue bars, and after treatment initiation, so that follow-up sample is represented by the purple bars. The days in treatment, again, are listed above the bar, and the lighter purple bars with an asterisk represent patients that were still culture positive at follow-up. So we evaluated the difference between the amperometric signal before treatment initiation and at follow-up. And this delta or change in signal was plotted against the number of treatment days in the bottom figure, with separate slopes for patients that became culture negative and those that did not culture convert. So controlling for follow-up culture status, days on treatment, we found that days on treatment was significantly associated with the decrease in CFP10 signal bilinear regression, suggesting that CFP10 reduction could be correlated with treatment response. Again, these results are very, very preliminary. However, they do suggest that this assay may have some utility as a treatment response monitoring tool. So in summary, this proof of concept study was able to demonstrate that our TB assay prototype was able to accurately differentiate among presumptive positive individuals, patients with culture positive TB. Our assay was able to produce results in less than 100 minutes using ultra low volume unprocessed clinical samples, and could be read by an inexpensive assay reader. Our study's results also suggest that this assay may have potential as a treatment monitoring tool. So next steps for this assay would be to simplify the current protocol and shorten the incubation times, and also to redesign the microfluidics to have, in theory, all the reagents self-contained on the sensor. Of course, we would need to conduct a large-scale field study using both bacteriological and clinical reference standards to evaluate the performance of this assay among difficult-to-diagnose populations, including people living with HIV, individuals with extrapulmonary disease, and children. So finally, I just wanted to thank all of you for this opportunity to present our study results. And I wanted to acknowledge my fellow collaborators, in particular, Ava and Victor from Joe Wang's lab, and of course, Tim and Tony, who provided the clinical samples that we evaluated. I also wanted to acknowledge the CFAR, which provided a significant majority of the funding for this project, without which this project would not have been conducted. And we also I also wanted to acknowledge that we did receive some additional support from the UCSD GEM program and the Arnold and Mabel Beckman Foundation. Thank you very much.
Dr. Seifert, thank you for that really excellent presentation. Those are really quite exciting and promising results. While I'm waiting for questions to be deposited, let me ask you a couple of questions regarding the potential utility of this in the field, especially in low and middle income countries where diagnosis is the most challenging. What do you know about the temperature and stability, shipping stability characteristics of this sort of technology?
So that's a great question. So we only assess the stability of this assay at refrigerated temperatures for 30 days, as I showed. But Joe Wang's lab has used the same kind of substrate and technology, and I think they had much longer stability at room temperature. We didn't try it with our antibodies, adhered to the substrate, but they have done it with other the same kind of technology, and did have quite a bit of success with months of storage at room temperature. So hopefully we would see the same thing.
And in terms of the technical requirements, in terms of personnel expertise needed and electricity or other resources, what are the requirements?
So the goal is to actually simplify it a little bit more, because there is some pipetting that has to occur now, with the microdroplet on the sensor itself, and then a washing step, and then another droplet, and another washing step, and then the reader step. But if our plan, the engineers, I've submitted an R01 to hopefully be able to do this, but the engineers have a plan already to self-contain all those reagents, so that then it would just be more of like a blister popping. So you wait for the incubation, and you pop the blister, and then it rinses it off, and so it's all self-contained. So it could be in theory done by very low technical capability, and then the reader itself can be attached to a cell phone. So you could take the potentiostat, attach it to the cell phone, and then read out the response. So the idea is to make it a really simple test, using urine, for example. That would be really low tech sort of application.
Mm-hmm. And would this be a single specimen sort of arrangement, or is a multiplex or multiple specimen possible? In other words, what would the throughput rate be for one technologist, for example?
So we did find a lot of difference in performance when we use different specimen matrices. So we would probably have to design the chips for a specific matrix. So it would be just like urine chips and or serum chips, or something like that. So likely, unless we can overcome that technologically, it would be specific to the matrix that you're supposed to use.
Mm-hmm. And I noticed in your one of your data slides, the sensitivity seemed to be greater in urine and serum than in buffer. That is unusual for most assays. What's the explanation for that?
They spent a lot more time optimizing it. So there's some I'm not a nano engineer, so I have to defer to Victor and Ava for this, but they spent a lot of time with laying down of the capture antibody and there's some they called it fouling things that occur. So like the buffer itself then gets distracted essentially, and doesn't capture all the antibodies. So they optimize it for specific body fluids. And so that's why some of the sensitivity is a little higher, but they did spend a lot of time, trying to optimize for specific body fluids. We didn't have any success with saliva. Actually, we tested saliva too, but there was too much interference. So we weren't able to get a signal using saliva.
Right. And sputum would probably be even worse. So, and what is the projected cost of this sort of thing per test?
So the Dr. Wang, I believe that projected, so the major cost would be the antibodies themselves. So if you had a supplier for antibodies, you could reduce that cost, but the actual physical, the plastic substrate and the electrodes are less than $1 a chip. So it'd be really inexpensive. So the cost would only be from the antibodies themselves. And we can commercially, we purchase them commercially, so they are available. But I think that kind of the overall cost would be less than $5 a chip. And then at scale, of course, it could be even less. And then the potentiostat, the reader itself is, I think they're commercially available. You can buy them for $500 to $1,000. So in theory, it would be a really low cost at point of care.
Well, this is all very exciting. I hope it works as you expand it to other specimens and situations. Is there anybody out there in the rest of the world who has a question for Dr. Seifert and our very exciting studies? If not, I want to thank you and wish you the best of luck.
Thank you. Thank you so much.